Hi everyone, welcome to the explanation of the poster corresponding to the work we have been developing, which is entitled Synthesis of Silver Nanoparticles with Antimicrobial Activity from the Extract of the Parasitic Plant Psittacanthus calyculatus. This work was developed by a server, Carlos Omar Santana Hernández, my colleague Andrea Guadalupe Pérez González, as well as researchers from the National Polytechnic Institute, in conjunction with a researcher from the Center for Research in Optics. First, I will give you a brief introduction. There are some species of plants that obtain essential nutrients for their survival from other individuals, to which they adhere named hosts. On the other hand, the species that perform this practice are called parasitic plants and currently represent serious problems for ecosystems. Particularly in the center, west and south of the Mexican territory is distributed Psittacanthus calyculatus, commonly known as mistletoe, which parasitizes various tree species in the regions already mentioned affecting the fauna and flora. Due to the wide availability of biomass that supposes the widespread presence of mistletoe and because of the reducing capacity of the extracts of this plant, provided by some phenolic acids present in its composition, its use was proposed for the generation of silver nanoparticles through the technique called grease, which uses extracts derived from plant components. On the other hand, for the characterization of nanoparticles, techniques such as spectrophotometry are used to identify the characteristic plasmonic resonance of the material, transmission electron microscopy, from which images are obtained that allow analyzing the sizes and morphologies. In addition, to establish their stability, their surface charge is analyzed through the zeta potential. A fundamental part of the study of these nanostructures is their application, since there are currently several fields for it. But in the search to generate new alternatives for the use of antibiotics, the use of silver nanoparticles as antimicrobials has been studied. This characteristic quality derives from the ability of silver to act as a reductant. It allows it to generate intracellular and extracellular damage in microorganisms such as bacteria while maintaining a low tendency to develop resistance, minimizing health risks. Methodology. Three main methodologies. The first corresponds to the green synthesis, which began with the preparation of the extract, heating to the boiling point 20 grams of the crushed leaves with 100 grams of the extractor solvent, which for this particular case is ethanol of 96 degrees. Subsequently, the extract obtained removed large particles and impurities. With respect to the green synthesi, 50 milliliters of the aqueous solution of silver nitrate 5 millimolar were heated to 85 degrees Celsius, then 5 milliliters of the extract was added and left to react for 2 hours at constant temperature and stirring of 850 rpm. Once the reaction time was over, a solution containing nanoparticles and some other residues was obtained. For the characterization of the AGNPs obtained, the sample was subjected to a purification train that consisted of centrifuging the sample at 8500 rpm for 10 minutes, then the supernatant was removed and suspended the precipitate with distilled water by sonication for 10 minutes. This process was performed on 3 occasions, resulting in a colorless supernatant. With the washed samples, characterization was carried out in the Genway 6715 UVVIS spectrophotometer, the GEOL JE100 transmission electron microscope and its zeta potential in the Z-Sizer Nano ZS, Malvern Panalytical. Finally, to evaluate the antimicrobial activity of AGNPs, plate susceptibility assays were performed. It was based on 3 solutions of 100, 50 and 25% concentration of particles using distilled water as a diluent. Of these solutions, 40 microliters were placed on filter paper discs of 6 mm in diameter, making 3 discs per concentration, and letting them dry for 24 hours. These discs were placed in petri dishes inoculated with Staphylococcus aureus ATCC 25923 previously prepared according to the protocol of Kirby-Bauer disc diffusion susceptibility, along with a control disc that did not contain nanoparticles. In addition, gentamicin discs were placed in another box as a positive control by placing the boxes in incubation for 24 hours. After this time, the inhibitory halos generated were measured, making 3 measurements per disc with the analysis program, to improve the reliability of the data.
Results. From the characterization by UVVS spectrophotometry, a sweep from 300 to 700 nanometers was generated, with A, and of which a wide curve is observed with a global peak at 470 nanometers, a value close to that reported for silver which oscillates 420 nanometers. This difference was generated based on the variable sizes and morphologies present in the sample, which were observed in the images obtained by transmission electron microscopy that can be observed in Figure 4, and thanks to which it was known that the diameters ranged between 4 and 99 nanometers. However, most of the AGNPs were found in a diameter between 11 and 30 nanometers. Regarding morphology, spherical, cubic and irregular configurations were observed. Finally, when studying the stability through the zeta potential, the nanoparticles presented a surface charge of -37.9 millivolts, this being a high value, which is a sign of a strong stability which helps to avoid agglomeration. Once characterized, the AGNPs studied its antimicrobial activity, obtaining average inhibitory halos of 13.37 mm, 12.36 mm and 11.85 mm for concentrations of 100, 50, and 25% respectively. However, the largest inhibitory halo found was at 100 U of 14.5 mm, which is 2.5 mm below the halo generated by gentamicin. That corresponds to 17 mm, although with this value it is already sufficient to consider a inhibition by AGNPs. Conclusion. In the generation of a protocol for the synthesis to have studies on the composition of the extracts of leaves of P. calyculatus, allowed to adapt similar methods for the synthesis of AGNPs which decreased the number of assays necessary to obtain the nanoparticles. On the other using characterization techniques generates a greater understanding of the properties of nanoparticles. In this particular case, through UVVS spectrophotometry, a wide resonant peak was obtained caused by the behavior of light when passing through the sample. This derived from variations in sizes and morphologies which could be observed by TM. In addition, in relation to the value obtained from the zeta potential, it was determined that strong stability due to the intensity of their surface charge. The inhibitory halos generated in the plate susceptibility tests confirmed that the synthesized AGNPs have antibacterial activity, so they can be used as an antimicrobial.